Okay, so people will eventually come in. Anastasia, if you want, you can proceed.
I will start from where I stopped, yes?
Okay, yeah, that's good.
You want me to do
No,
it
no,
again?
that's okay, that's okay.
It's okay. Okay, thank you. Okay, so after this event, okay, you can say, let's continue. So our work here was to synthesize these interesting compounds using either protic ionic liquids or deep eutectic solvents. We were gratified to see that the reaction proceeded smoothly in both cases, although a little bit better and with lower temperatures using ionic liquids. So we could recycle and reuse the solvents up to 4 times. A very high atom economy, as expected in multi-component reaction, was observed, and also moderate to good yields were obtained with high purity compounds. So these compounds are of pharmaceutical interest because they showed anticandidial activity. So following that, we went on to develop a new methodology on the Knevernagel condensation reaction using proline-based NADES to synthesize orones. Orones are natural pigments. They are minor flavonoids responsible for the bright yellow and orange colors on ornamental flowers like the snapdragon that you see here. And they possess a wide range of bioactivity. The thing with their synthetic roots is that they demand mercury acetate and pyridine as reagents, or strong acids or bases. And an important feature also is that in both green, to make this process more green, what we did was use proline-based NADES. Proline is one of the first organocatalysts that was applied in various organic syntheses. So by synthesizing proline-based natural deep eutectic solvents, we could recycle and reuse the solvents up to 6 times without loss of catalytic activity. Good to very good yields. And the most important thing, we didn't require the phenolic hydroxyl groups did not require protection and deprotection, and no other purification step was required. So after that, we proceeded to develop green extraction processes using NADES. So the problem in industrial processes using extractions is that they use high amounts of organic solvents, and this causes and this is energy consuming, and it causes volatile organic compounds emission. So and also some toxicity issues arise because of the traces of organic solvents that might remain after the evaporation. So one potential solution is the use of alternative solvents, such as natural deep eutectic solvents. These can be task-specifically designed so that we can select the starting materials to be generally regarded as safe. Make sure that they are currently used in the applications that we want to use them to. And apart from being effective extraction media, these NADES can act as protective carriers of the extracted bioactive phytochemicals and provide synergistic activity. And moreover, they can act as plasticizers in various biocompatible materials. So an additional advantage is that we have 2 ways to exploit the extract. One is the traditional way, we could say, the recovery of phytochemicals by liquid-liquid extraction, solid-liquid extraction, and anti-solvent addition. The other way is to use the extract as obtained, meaning that we can As we said, the green solvents can serve as protective carriers for the extracted compounds, and they can help with the bioactivity. So we just choose them carefully, and we use the extract as obtained. So in this context, we have developed a lot of different extraction processes using NADES for the valorization of agri-food waste or plant material. And then what we have been up to is to use these added value extracts for the development of novel end products such as cosmetic formulations, biocompatible films, and hydrogens. So one interesting application of this development, green extraction development, is the agri-food waste valorization. And we have been working on this for the past 2 years for the development of a green methodology for the extraction of olive leaves. Olive leaves are a byproduct of farming of the olive grove, and they consist around 10% of the total weight of the olives. And it is a nice raw material because it is of low cost and high availability all year round. And it contains high amounts of oleuropein and hydroxytyrosol, which are very important bioactive compounds. So this is a scheme to show what the procedure that we follow. So we first have our dry olive leaves, and then we do a screening on different NADES, and we use ultrasound assisted extraction. And after some preliminary experiments, we optimize the conditions of the process using design of experiments. And then the added value extract that we obtain is characterized and evaluated for its bioactivity and cytotoxicity. So just to show you that we have been monitoring the quality of the extract using HPLC, and by seeing how much oleuropein and hydroxytyrosol we have obtained, as well as measuring the total phenolic and total flavonoid content. We chose the glucose lactic acid as the best extractant because it combined all the qualities that I just mentioned. So after optimization, we went on to find the optimum conditions, and we also performed an extraction using conventional heating at 40 degrees for 3 and up to 3 hours. However, we did not manage to obtain an extract with the same quality as in the case of ultrasound extraction. So the NADES, as well as the extracts and the pure oleuropein, hydroxytyrosol, and tyrosol, were assessed for their cytotoxicity. And this was partly work done by Dr. Andromachi through her STSM in September 2021 at Nova School of Science and Technology and DES SOLUTIO company. We were very gratified and happy to see that our extracts did not had a very good cell viability in 3T3 cells and Hackett cells, and this gives us the opportunity to believe that they can be exploited further because they're not cytotoxic. So a second material that we are working on is the extraction of wild rose hip. This is a very nice plant, and the rose hips are rich in ascorbic acid and valuable phenolic compounds, and the extracts are used for cosmetic formulations. So we set out to develop an innovative green process which comprises of 2 parts. The first one is enzyme pre-treatment of the material, of the plant material, and then extraction with NADES. And here comes the difficult part, because the final task, the final aim of this project is to incorporate the extract in cosmetic creams that are going to be used by Kore's natural company, natural products company. However, they have a lot of restrictions. They do not want urea. They do not want very acidic NADES. They do not want choline. So there are a lot of different restrictions that you have to be careful of before you select your NADES. So after doing a small screening, we ended up with beta-glycerol, which is okay with the cosmetic industry, and it is not very expensive. Another example of agri-food waste valorization is the extraction of brewer's spent grain using NADES. This is a very important and common byproduct of the brewery industry, and it has a lot of phenolic compounds. And again, we did the same, let's say, we followed the same path. And but at the end, the beta-glycerol extract was incorporated in cosmetic creams. So we did this, and we did all the different tests that are commonly done in these types of formulations, like the freeze-thaw stability, the ultracentrifugation, and we tested the stability of the formulation and we saw that it was stable for 4 months. The texture was fine, the color did not change. So again, it was a very promising, let's say, application, this one. We are very happy that our work is starting to get acknowledged by the industry. And some months ago, we took part in a contest, an idea, talent, sustainability, organized by Friesland Campina Hellas, and we were awarded the consulting support from the company in order to exploit the cheese whey, which is a main byproduct of the dairy industry, in order to make new protein films or protein rich extracts in order for them to exploit them in high protein products and protein films as coating. Because NADES do not stop to make us creative, let's say. We have used them as plasticizers for the preparation of chitosan stabilized films. So these films were prepared using solutions of beta-lactic acid NADES and choline chloride lactic acid NADES. And by dissolving chitosan in these, we were able, after evaporating the water, to form nice films. You see here the same images of these films, which had very interesting properties, such as enhanced water vapor permeability in comparison to the films that were prepared using acetic acid to dissolve chitosan. And this could be very nicely exploited as wound healing materials. And now we are working on using NADES in hydrogels preparation. So hydrogels are 3D hydrophilic polymeric networks. They do not dissolve in water, but they can swell and retain water for a long time. And they are classified using different, depending on their sources, pore size, nature of swelling, etc. So in this context, what we did was we just wanted to work on some materials to use as food supplement and weight control agents. So we decided to combine chitosan, which is a well-known biopolymer that helps weight control, essential oils that also do this, and alginate to make hydrogens. So what we did was encapsulate the essential oils in chitosan, which was dissolved also in NADES solutions, aqueous solutions, and then we performed the final cross-linking using alginate and calcium chloride. So we were happy to see that the new materials that we produced had a very high swelling ratio, up to 460%, and they retained water almost for 2 or up to 3 hours without a considerable loss. Finally, what we do now is try to incorporate the nice olive leaf extract that I told you about a few minutes ago in chitosan hydrogels. So here what we do is just use NADES and nothing else for the preparation of the hydrogels. So no need to add other cross-linkers. And this is a very good opportunity to develop even greener materials. This is a work in progress, but still we can see that we have a good swelling ratio and a good retention ratio. So our scientific journey is going on. We have the scientists, the facilities, the ideas. Of course, there are a lot of hurdles in the way, and one of them is how to find the way to let's say, to communicate with the industry. However, I am positive, I'm sure that these types of webinars and collaborations are now paving the way towards these collaborations. So I will thank you very much for your attention, and I will close with a nice photo of our campus covered with snow from the snow storm that hit Athens 2 or 3 days ago. This is something very not common for us. So this is why we think that it is good for you to see. So thank you very much.
Thank you, Anastasia, for your very nice presentation. I would say that maybe we would continue with Alexandre's presentation, and we would leave the discussion for the end, if that's okay with you. Yeah. Okay, so
let me stop sharing.
Yeah. Can stop sharing, and then meanwhile I introduce you Alexandre PAIVA. He's a researcher at Nova School of Science and Technology, but he's also the CEO of DES SOLUTIO, a spin-off company from Nova, which is devoted to the development of natural deep eutectic systems for different applications. And so without further ado, Alexandre, the floor is yours.
Thank you so much, Rita. So welcome everyone, now that we are back. What I'm going to talk today about, I wanted to show you after, because Anastasia already talked so much about the part of extraction and what we can do with the implementation of deep eutectic solvents. I wanted to give you more a little bit the perspective of a company that me and Rita started. That is DES SOLUTIO. And why did we do this? Well, what happened is that we started realizing that we had a lot of interest from industry that came to us and asked us about deep eutectic solvents, how they could incorporate deep eutectic solvents on their products. And most of these companies were actually from cosmetic industry. This was mainly due to the restrictions that are increasing more and more, especially in the European Union, restrictions about the ingredients that can be used in this industry. And so the companies are looking for alternatives to this, and they somehow discovered that deep eutectic solvents may be maybe the answer for this. And that's why we created DES SOLUTIO as a way of trying to bring these solutions to the industry and trying to develop a little bit, because we realize that there's a potential for deep eutectic solvents to be implemented into industry. And this is the idea here is what is to always originate green-based solutions. So NADES can have a lot of potential in creating these great green-based solutions, starting from natural products, either for the production of the deep eutectic systems or as feedstocks for creating extracts, natural extracts, using DES as the solvents. And as I will show you later, the big advantage here is that, and Anastasia already talked a little bit about this also, the big advantage here is that these deep eutectic solvents are not just like common solvents that we use and then we need downstream processing afterwards to obtain our extract. So they can be bioactive extracts, and we can have a bioactive deep eutectic solvent with or without an extract. And this opens a lot of possibilities for the creation of new formulations for nutraceutical, for cosmetic, pharmaceutical, and even biomedical and personal care. But there's still a long way to go until we are fully implemented into this industry. And in our point of view, the big challenge that we have today with deep eutectic solvents is in there's no rational design so far for the formation of deep eutectic solvents. So we have, for each solution that we want to find, we create a deep eutectic solvent specially designed for this. And this is done based on trial and error and past experience. Of course, that we have experience that we know that this eutectic system worked for that application, and we use this knowledge to create new ones, but there's no rational design. This is time consuming, and most important, doesn't allow us to quickly design a solution for a specific problem. And the second problem with this is that we can also not predict the properties of the deep eutectic systems. We all know that, we all that work on this area, we know that the big problem with deep eutectic systems is the viscosity. But there's also, and of course, if we have a solution for a product that has to be injected, for example, it cannot be cannot have a high viscosity. And even for extraction, if you want a deep eutectic solvent that can be used for extraction, if it has a high viscosity, of course, we will have problems in the scale up of the process due to the mass transfer problems. Of course, acidity is also a problem. Many of these tests, especially if we are using choline chloride, they have a problem of high acidity. And this, of course, can transpose these problems then for toxicity and biodegradability, especially toxicity. If it is very acidic, I can extract a lot of the component I want. I have a deep eutectic solvent that has a high solubility for antioxidant A or B, but then it has a high acidity, and then I cannot use it on a formulation. So this, I think computational tools will be the answer for this, and this is something that, of course, there's a lot of work going on this subject, especially because of this, because then this will all be reflected in the final product and on the price. But there are already, as we all know, there are already NADES implemented in this way. The big examples are Eutetics by Naturex, that was recently bought by Givaudan. And of course, Gat4C, that has already some extracts, natural extracts with deep eutectic solvents that are using in cosmetic formulation. So, of course, NADES already are in a point that can be used in the industry, but there's a lot of fundamental research that needs to be done so that we have a more rational design towards the starting point and the implementation in the industry. But the deep eutectic systems are truly the sustainable approach. And if you do a kind of a benchmark regarding the usual suspects, the competition, the highest competition that are other green solvents and of course organic solvents, deep eutectic solvents have two main advantages in our point of view. The main, of course, they are biocompatibility, because they are based from natural products, normally natural metabolites. They are biocompatible. And this is, of course, the main advantage. But the biggest advantage in terms of implementation, and why deep eutectic solvents, I think, had such a fast implementation in the industry regarding these other solvents like ionic liquids and supercritical fluids, is that is the simplicity of their production and the simplicity of the products that we create with deep eutectic solvents, because there's no need for downstream processing. Because they are biocompatible, and because some of them even have bioactivity, they can be used directly as a final product, as a final formulation. So we don't need a downstream process to obtain the extract and then formulate it again. This is a big advantage. The process is simple, much more simple. The cost is much lower and more important. I don't obtain a few grams of my extract, I obtain liters, because the production is much higher. My product is not just the extract, is the active solvent. So it's the deep eutectic solvent with the added value of the extract. This is a big advantage when implementing, in this case, more directly extracts into industry. I wanted to show you a little bit these four pillars, for topics that we are in DES SOLUTIO, we are working more, and where we are a little bit more advanced in terms of implementing these solutions into industry, and where we are already on the, well, I don't want to say fast track, but at least in a good way to implement this into industry. And these are the four markets that well, there are a lot of markets where deep eutectic solvents, luckily they are very versatile and they can be implemented in a lot of markets, but these are the ones that we are focusing more, mainly due to our expertise, of course. Well, extracts, as I told you, I cannot talk a lot about this because of NDAs, but where we started working, developing more the deep eutectic solvents, and I would not go much into detail, and Anastasia already went into detail regarding these extracts and show you how they can be valuable when we are talking about extraction. But is the valorization of agroindustrial byproducts, transforming them into cosmetic formulations, where the deep eutectic systems are used both as both as extraction solvent for these byproducts, for many agroindustrial byproducts, and then can be incorporated in formulations. Of course, the trick here is that we must have a stable formulation with activity and, of course, with all the properties that cannot damage the skin. In this case, we are talking about topic formulations. Another interesting market, and just to go away a little bit from extraction and nutraceuticals, because deep eutectic solvents, we always talk about that we are inspired by nature. What we were inspired here was looking at animals that survive environments with a very high amplitude in temperature. For example, in Alaska and so on, when you can have winters with minus 20 degrees or minus 30, and then we have summers with 25 or 30 degrees. So how do animals survive this? What we realize is that in winter, for them to survive, they literally freeze. And afterwards, in spring, they start to warm up and then defrost and completely survive the process. How do they do this? How do they survive the dehydration and the ice crystal damage in the cells? Well, they do it by producing metabolites during autumn and winter to avoid when the weather starts getting drier and colder. And they do this by producing these metabolites that avoid ice injury and dehydration of the cells. And if we look at these trehalose, urea, glucose, glycerol, and so on, these are components that we use normally in when producing deep eutectic systems. And we thought, why not try to produce a deep eutectic system that can combine the advantage of more than one component, one metabolite. And we hypothesize maybe the animals use these hypothetical systems to survive. Why not use these for cryopreservation of cells? And this is what we've done. And we've created NADES, which is a solution that we now already have developed. And what we realize is that it has several advantages. Normally, cryopreservation of cells, what is used is DMSO, that is the golden solvent, that although is it has a very high efficiency, it is toxic for cells, so it has to be removed before the cells are injected into the patients. We are talking about, for example, cell therapy. If we talk about cell therapy, that is considered almost the future of medicine because of the therapeutics for chronic diseases, where you have these stem cells that must be stored somehow before being injected into the patient. This is done using DMSO, which means that we have to freeze it in liquid nitrogen at minus 196 degrees. Then we have to defrost it, remove the DMSO before we can inject the cells into the patient. This is very expensive, this is very complex. That's why cell therapy is not accessible for everyone, and it's a very expensive therapy only for a few people that can use it. Deep Eutectic systems have the advantage of having the same efficiency, as you can see here, the same efficiency as DMSO. So the cell survival after freezing and defrosting is the same as DMSO, but the toxicity is much, much, much lower. So we have the efficiency of the golden solvent for cryopreservation without this toxicity. What this means is that there's no need for complex processing of the cells before injecting into the patient. And the biggest advantage also is that we did some experiments to realize that this efficiency is at minus 196 degrees when freezing cells at this temperature, liquid nitrogen, but also when we freeze it at minus 20. Minus 20 degrees is the temperature of a normal freezer. So this completely simplifies the cryopreservation process and allows for reaching completely new markets as, for example, cell therapy, not just for humans, but for example, if you think about animals, cell therapy is started being used in animals such as race horses and so on, but veterinarians don't have the access to this technology to freeze it, liquid nitrogen and so on. So this can open new markets, and this is one example where deep eutectic solvents can reach very interesting, very big markets, and where it can be a major play, changing a little bit the status quo of this case cryopreservation when only DMSO is used. Another example that we wanted to show you is we started working with these hydrophobic deep eutectic systems, based on menthol and fatty acids. And what we realized, well, we know that many deep eutectic systems have antimicrobial properties, but what we realize also is that they can avoid biofilm formation. Biofilm is basically the way that bacteria is used to ensure their survival. When they start growing, they start producing this biofilm that is produced by the bacteria and allows them to protect themselves from the external environment. So they create this biofilm where they start growing, and then they are protected. And this allows them to grow much more efficiently and much faster. If you can kill the bacteria, but if there's some bacteria survival, then they start growing again because they are protected. If you detach this biofilm where the bacteria are growing, then they cannot attach to the surface and then cannot grow. Why is this? You can see here, for example, with 3 different bacteria, that when we apply the deep eutectic solvents on a colony of bacteria, what happens is that you clearly see a decrease of the population, and it's decreasing very fast. When without deep eutectic systems, they grow without any problem. This is why, because they cannot adhere to the surface, and so they start dying because these hypothetic systems are antimicrobial, so they kill the bacteria, but more important, they don't allow them to attach. Even if they don't die, they cannot grow. And this has very important industrial applications. And this is why we are working towards this, because not only social, because bacterial infection, and if you are talking about the healthcare, for example, 7% of all patients that 7% of all patients hospitalized acquire some sort of healthcare association infection. This is due with normally, unfortunately, with resistant bacteria, which is very hard to kill. And for you just to have an idea, one out of 70 patients in the United States that acquire these infections, they die. So this has a very immense social burden, but also economical. This has a cost of about $36 billion just in the US. So this is a huge market in the healthcare, but also, for example, just to give you, we all seen the sheep holes that are completely full of this macrofolding, these animals that are attached to the hole. And this can result on increased drag of the ships, which means that they will spend about 35 to 50% more diesel doing their trips, and have a huge cost on the companies. So if we have a solution to avoid the attachment of this bacteria, if the biofilm does not form, bacteria do not attach, and the other animals cannot also attach to the sheep hole, for example, or even to the medical devices that can be be subjected to infection by bacteria. So there's a huge market with a lot of not only potential, but also importance, social importance, and that can be explored. And hypothetic systems here, because once again, they are biocompatible, okay? And also we are working a lot of understanding the ecotoxicity of the hypothetic systems, because if you are always claiming that they are biocompatible, that they are very green, we have to know the ecotoxicity. And if you want to use it, for example, in sheep coatings, well, if they leach to the ocean, they cannot cause problems. And what we have realized that the ecotoxicity of these hypothetic systems is very low. So there's once again a market that can be explored where the hypothetic systems have a lot of potential and can be game changers. Another interesting where we have been working for a couple of years, and we are developing some products, is also in the biomedical industry. And we started working with hypothetic systems and combining compounds. When we are combining and designing the hypothetic systems, trying that one of the components is an active pharmaceutical ingredient. So that the hypothetic solvent itself, the hypothetic system itself, can have therapeutical properties. We call it TDES, that is therapeutical hypothetic systems, because, well, this is exactly what they do. And we started working well in a lot of applications, but one that gave us a lot of attention when we realized that there were a lot of potential for the hypothetic systems was exactly in cancer treatment. Well, in this case, terpenes are known as anti-cancer agents that kill cancer cells, and also what happens is that in the development of and the multiplication of cancer cells, this is an inflammatory process. So anti-inflammatory drugs such as ibuprofen and so on can reduce the multiplication of cancer cells. So they can help preventing the proliferation of cancer cells. So if you combine a terpene such as limonene with an anti-inflammatory drug such as ibuprofen, this may be a good solution. It can be a drug effective against cancer treatment. And we did this against CACO2 cells that are cancer cells and measure the cytotoxicity in keratocinites. And what we realized is that these TDs, limon and ibuprofen, was more than 4 times more selective towards cancer cells. So it killed much faster cancer cells than normal cells. This you can see here the EC50. The lower means that there's the lower concentration that you need to kill the cells. So it is much more active towards killing cancer cells than killing normal cells. So what we don't want, cannot be toxic. So what we have here is a treatment, a drug that is much more, is very effective towards cancer cells. But this is not just the big advantage that we have here. We have a higher advantage that is also the solubility of these drugs. So what we realize is that when we have the solubility of ibuprofen, for example, in water is very low, but if we have it in a deep eutectic system, when we dissolve the deep eutectic system in water, in a PBS solution, of course, but when we dissolve it in an aqua solution, we realize that the solubility of ibuprofen can be 4 times higher than pure crystalline ibuprofen. This is a very interesting thing that we realize, because this increases the bioavailability of the drug. So it is absorbed much faster, and we need much lower concentrations of the drug to have the same effect, because the solubility is higher. So what we are doing basically is turning an API, a drug that is type 4, which means that has a low solubility and a low permeability, in a type 1 drug that has a high solubility and high permeability. So this means that we can design a number of drugs that can be much faster acting, and we need much lower concentration. So we don't need to take a lot of a big amount of a drug just so that a small amount can have a therapeutical effect. So basically, the message, we are a little bit late. So the message here that I wanted to leave to you is that the implementation of deep eutectic solvents, although we still have a long way to go, so that we have a systemized process for designing solutions towards different problems. We already are doing it, and we are doing this by designing specific deep eutectic systems and studying these specific systems to design new formulations. And the applications and the markets are willing to adopt the solutions. We are talking about anti-cancer formulations, antioxidants, cosmetic formulations. The cosmetic industry is very keen of these new solutions. And so there's a big market for deep eutectic systems that we realize this in what we are doing now as DES SOLUTIO, is trying to push and implement these deep eutectic systems in this industry. So thank you so much for your attention, and well, if you have any questions.
Thank you, Alexandre, for your presentation. Maybe, yeah, you can stop sharing the screen, and now we open the floor for discussion. So if anyone from the audience would like to ask something to Anastasia or to Alexandre, please feel free to do so. Okay, well, we still don't have any questions from the audience. So I have a question for you. Why do you think that the cosmetic industry is ahead in this field in the use of eutectic systems in their formulations? Is it because the regulation is less strict? Is it because of the advantages that these systems have over others? Why is this happening?
Well, in my opinion, is a little bit with the legislation. that of course is less strict than the pharma, but in the other way, it's there's a problem that in the case of European Union, most of the ingredients that were normally used in cosmetic formulations are being forbidden and are being considered as toxic, and most of them were widely used, so they need solutions for their formulations. And also the other possibility is that it's a very competitive market, so it's you always need new products, you always need new marketing strategies so that you are in front of your competition. So I think this is why the cosmetic market is so much more active than the other industries.
And what is your opinion on what are the next industries to follow this? I believe, well, maybe pharma will be one of the last due to the regulation that there exists, but in other areas, what's your opinion?
Well, I don't know if Anastasia wants to say something, because I was going to say food industry, but Anastasia knows more about it than me.
No, I totally agree with you as far as the cosmetic industry is concerned. Also, I would put the scale up, which is quite easy with deep eutectic solvents. You can count on it, okay? So they will not have a problem with that. But for other industries, what I see is the biomedical industry, maybe where we can apply deep eutectic solvents for new materials and all that, because of the problems with the regulations with the pharma, I think. Although those applications that Alexandre just showed are amazing, so I think you can easily convince pharma to for the APIs and for the solubilization of APIs. I think this could be, but if you consider, let's say, to enter some greener steps in a synthetic process, let's say, for an active pharmaceutical ingredient, there you see a very strong opposition from the pharma. It's very difficult to convince them.
Okay, so in fact, these systems are not, well, they used to be named as the deep eutectic solvents, but in fact, they are not only working as solvents, and as Alexandre said, they will be part of the ingredient, the final product that it will use. Maybe it's not going to be the same in all areas, and in some others. Maybe this can replace organic solvents in polymerization reactions or in other organic chemistry reactions, but this, I guess, is a little bit much further from the industry than the examples that you have shown here.
Yeah, because we have some experience, for example, as using in biocatalysis, and realize that this hypothetic system has the ability to stabilize enzymes, for example. So in pharma industry, where biocatalysis has some importance, this can be an application. But as you said, it's true, we are much further away from industry than we are, for example, for extraction, for the formulation of new therapeutic systems and so on.
Okay, I don't know if now somebody from the audience would like to add something or some comments or questions. If not, then I'm going to thank again Anastasia and Alexandre for these lectures. I'm sorry again for all the problems in the first part, but now everything was fine. And I hope to see you soon in Coimbra for the joint meeting of Greenering. And otherwise, we will meet in the next webinar. Have a nice day and have a nice weekend. Bye bye, everyone.
Bye.
Bye bye. Thank you.
Bye, everyone.
Bye.
Bye.